Right, so today we're going to be telling you about the Brain Observatory, the Allen Brain Observatory, which Matt just told you is our survey of 2-photon calcium imaging data collected throughout the mouse cortex. And in previous years, there have been team talks that have detailed the ways in which we built the standardized pipeline from members of our neurosurgery team, our OPHYS core, our engineering team, the methods that are being used to collect this data in a highly standardized way that are very robust and allow us to use a lot of quality control metrics to make sure that all of this data is very powerful and good to use. There was also a talk a few years ago from members of our informatics team about the ways that the work they've done to kind of integrate the different pieces of data that come out of this pipeline in order to create our data product, which is a website. We've got some screenshots here showing the types of visualizations of data and lots of metrics that are being computed. Today, what we're going to talk about isn't how we collected the data or created the product, but some of the things that we're learning from this large, powerful data set and ways that we're building on it and moving into the future. So since the first release a little over 2 years ago, at our most recent data release in October, the size of this data set is over 60,000 neurons that have been collected across 6 different parts of the mouse visual cortex using a wide range of different transgenic lines, 16 different transgenic lines in total. And we're trying to characterize and look at how the visual information is being represented and transformed throughout this circuit. And this type of work really stems from kind of pioneering work by Hubel and Wiesel. I think many of you are probably pretty familiar with these types of experiments. What you're seeing is the stimulus being presented to a cat while they record from a single cell. You should be able to hear some spiking activity. So the spiking activity, they have a single electrode recording from a single cell. And they move the stimulus around in different places, and they start marking up the screen to indicate the regions of space that drive activity from this cell, which they've marked here with these X's, and then other regions where we actually hear a suppression of activity flanking that. As they do this, they create a description of this cell's receptive field. You see here these X's and these open triangles showing these suppressive flanks. And this provides a description of the cell's response, right? We can put some metrics on this in terms of the location of space relative to the cat's gaze, the orientation of this bar that most effectively drives the response of this cell, the general size of this area that drives activity, the actual width between these things. We can put some metrics on it to describe the response. We can also want to take this a step further to build a model of the cell's response. And this little cartoon drawing of X's and triangles actually serves as such a model. We can make predictions of the types of stimuli that will continue to drive activity from these cells and the types of stimuli that won't. And you can consider the rest of their experiment here as testing those predictions. They move this bar at different orientations. At this orthogonal orientation, there's no response because the excitatory and the suppressive areas cancel each other out. But when it's nicely aligned, we actually hear pretty robust responses where that bar of light falls nicely onto this model of the receptive field. So this is what we're trying to do with the Brain Observatory data is describe the responses we're seeing and then also build predictive models that we can use to better understand these representations and their transformations. This illustration here, so from Hubel and Wiesel's work, they found roughly 2 types of responses in the cat cortex. This is kind of a different version of those X's and the open triangles, an area that drives activity and areas that kind of suppress the activity, where you can kind of linearly predict the responses based on the summation of the patterns of light that fall into these different subunits. They also found another type of cell where you could not make that type of linear prediction, where the excitatory and suppressive areas were superimposed with one another, overlapped. And so the cell still responded to a bar of light, but it responded to an oriented bar of light in lots of different positions, and you couldn't make that linear prediction. And Hubel and Wiesel called these simple cells where you can make a nice, easy linear prediction, and cells that they don't understand and hence are called complex cells. Now, as I mentioned before, in the Brain Observatory, we are conducting this large-scale survey in order to characterize these responses in the mouse visual cortex, and we designed our experiments in order to survey across multiple visual areas. We used a large battery of different transgenic lines, both excitatory cells and inhibitory cells, that also get us expression in different layers, even into deep layer 6, as well as with some Cre lines that drive the expression of our calcium indicator in specific subpopulations within a single layer. This allows us to look at different cell types as well as different layers of the cortex. And finally, we're using a very large set of different stimuli. In Hubel and Wiesel's experiment, they could hand-tune the stimulus for each cell based off of the responses they were seeing. And we clearly can't do that in these experiments. So we use a wide range of stimuli and the same set of stimuli for all of our experiments. So we have a very standard set of data. The stimulus set includes some artificial stimuli, such as these gratings and sparse noise stimuli, as well as natural scenes and natural movies. This is just an example of one of the experiments. This video was of the raw data that Jerome's team has put together. So we're driving the expression of GCAMP6 in the population of cells using our Cree lines. And so you see, instead of recording from one cell, we're recording from a population, usually about 200 cells. And instead of hearing those spikes over the speaker, every time a cell responds to the stimulus, we see that it lights up. At the same time, we're presenting some diverse visual stimuli. You can see what was on the monitor over on the far right. We're also tracking the mouse's eye position, which we've superimposed here on the stimulus. So you can get a sense of how much the mouse moves its eye, which in general isn't very much, but it does actually move a little bit. So just to give you a sense of the type of data that we're seeing, here's one example cell. This is an excitatory cell in layer 5 of V1. We can look at its responses to all of our different visual stimuli. So in response to drifting gratings, these are gratings that move in 8 different directions and at 5 different speeds. This is one of the visualizations from our website, and we see that the cluster of red dots indicates where it's responding, which in this case corresponds to horizontal bars that are moving upwards at kind of these intermediate speeds here of about 2 or 4 hertz. In response to our flashed static gratings, again, these are flashed at different orientations, at different spatial frequencies, different widths of the bars. And again, we see that this cell responds, again, to the horizontal bars at these kind of, again, intermediate widths, not too broad, not too fine. In response to our locally sparse noise, this allows us to characterize where on the monitor is the cell responsive to. Where is that receptive field located? We're able to map a receptive field for both the white squares and the black squares separately, so we can look at these different subunits independently. This particular cell only responds to white squares kind of off in the left-hand side of the screen, so the equivalent of this region of space relative to the mouse's viewing. In response to natural scenes, this particular cell responded to about 4 of the different natural scenes. And we can look at its response to our natural movie. Here, the response kind of goes around this visualization as a circle, and it's a fairly sparse response, so you can't see very much, but you can see across the 10 different presentations of this movie, there's 2 little time points here where we see a pretty reliable response across these 10 different trials. So, as I mentioned, we can quantify, we can put metrics to these responses in order to characterize them in terms of the location and the area of the receptive field, or the preferred orientation of the gratings, or how selective the cell is for direction as opposed to simply just the orientation itself, the reliability of these responses across trials. And so we've computed metrics like these for all of the cells in our data set, and we can start to then ask some questions about how these vary across our areas and across our layers. And so let's now consider whether we see any differences across the 6 cortical areas that we've collected data from. And I'm going to represent some of these data using what we call our paw plot, where we are representing data from each visual area with one of these circles roughly aligned as they are on the surface of the cortex, so V1, and these 5 higher visual areas around it. Because we've collected data from multiple layers, we will have one of these for each of the layers we've collected data from, where the color now is going to be used to represent the average metric for some of these derived metrics. So here I'm showing you the direction selectivity of the neurons. And this is how strongly a cell prefers direction in one motion versus its opposite motion. So a cell that only responds to gratings moving to the right will have a very high direction selectivity, whereas if a cell responds to gratings that move to the right as well as gratings that move to the left, it will have a low direction selectivity. What we see when we compare across the different areas and the different layers is that across all areas, the cells in layer 4 have the highest direction selectivity. And this is particularly true for V1, where we have the highest direction selectivity across all of the areas, all of the layers here in V1. We do see some evidence that there's differences between some of the higher visual areas in terms of their sensitivity to direction of motion, and that these 2 lateral areas, AL and LM, show higher direction selectivity than these medial areas, AM and PM. And we see these differences primarily in layer 4 and in layer 2-3. In the deeper layers, however, the differences between areas become non-significant, where we see that not only are the cells in general less direction selective, but the differences between the areas are no longer there. We can put this, compare this particular metric, the direction selectivity. This is a single cell metric that we've just averaged across all of the cells in these particular areas and layers. And I'm going to compare it to a population metric where we're looking at how well we can determine the direction of motion on a given trial based off of the activity of all of the cells in that particular experiment. And what we see here is that the decoding performance, how well we can predict the direction of motion, really nicely mirrors the single cell direction selectivity, the average direction selectivity for these areas and layers. So we see the best decoding performance here in layer 4 of V1. We see that these lateral areas in the superficial layers then do a better job than the more medial areas. And then when we get into the deeper layers, that not only is the decoding performance somewhat worse, but the differences between the areas are no longer very significant. We can then add a layer of detail by pulling out different Cree lines. In these paw plots, we've only shown a single Cree line per layer that we've sampled really nicely across all of the areas. Here I'm showing summary metrics within V1 for all of our Cree lines in our data set. And we generally see, again, that this correspondence between the average direction selectivity for a cell type corresponds to its average decoding performance, with some exceptions. And so, for instance, this particular layer 4 Cree line NR5A1 shows the strongest average direction selectivity, but actually has the worst decoding performance among all of the different Cree lines in V1, which raises some interesting questions about how this population might be functioning differently from some of the other populations. So this is an example of the type of characterizations we can make of the response properties that we're measuring. But now we want to move to this question of, can we build predictive models of the responses that we're measuring? And so Michael Oliver is going to tell us about the work he's been doing developing these models.
So we want to build predictive models that reflect our current understanding of visual processing. These models can be seen as instantiating a hypothesis about visual processing. Then we can test our hypotheses by seeing how well these models generalize to new data. So we're going to model the Brain Observatory data, and we're going to model the stimuli, the natural stimuli, separately from the artificial stimuli. So we're going to look at the natural images and movies together, and then the static gradients, drifting gradients, and locally sparse noise together. So we can see which type of stimuli allows us to create the most predictive models. And so we're going to build a predictive model that takes these stimuli as input and then output a predicted response. And we're going to fit this model to one set of data and then test it on a separate set of data. So the model that we found that is currently most successful for us is one based on the Gabor wavelets that Saskia mentioned. And these are three-dimensional Gabor wavelets. So we're modeling not just the spatial receptive field, but also how that evolves over time. And so we have two sets of Gabor wavelets. We have a linear set, so it's just the output of the wavelet. And then we have a quadratic set, which is the output of the wavelet squared. And so the quadratic ones are used to model complex cells, and the linear ones are used to model simple cells. And the model for each neuron is simply a weighted sum of the output of all these Gabor wavelet features. And the weights are found by a sparse linear regression. Also included in the model is the running speed, since some neurons are known to be modulated by running. And Dan is going to talk about that in just a second. So once we fit the model for a given neuron, we can then look at its predicted response on a separate set of data and see how well our model predicts that response. And we can look at that in terms of correlation. And so when we do that, we see that the models trained on the natural stimuli create far more predictive models than those trained on the artificial stimuli. We see that we do have pretty high correlations for many neurons, but there's still quite a few for which our correlations are not as high as we like, which for reasons I'll get to in just a second. And once we fit these models, we can then try to interrogate them to learn more about the processing that is going on in each neuron. So one thing we can look at is to try to quantify the complexity. So to do that, we can create a complexity metric just by looking at the weights on the quadratic wavelets relative to the weights of on all of the wavelets. And so this metric will be 1 if only quadratic wavelets are used and 0 if only linear wavelets are used. And we see that even in V1, this metric tends towards around 0.9 for most of the neurons. And the regression selects many more wavelets than we might expect, indicating there's something more complicated and nonlinear going on than we might have initially thought. So the other problem is that models are data hungry, and we see that there's a very clear relationship between the number of events we collect for each neuron and how well the model predicts, indicating that our models might not be sufficient, and also we might not have enough data to fit even more complicated models. However, for a comparison, this Hubel and Wiesel experiments that Saskia mentioned were based on about 20 minutes of data collected using very simple stimuli. The Allen Brain Observatory collected about 3 hours of data per neuron using a variety of stimuli. And we're currently working to collect a set of data of 10 hours of natural movie data for each neuron, which we'll then use to try to fit more sophisticated models. So one such type of more sophisticated model is based on convolutional neural networks. So a convolutional neural network learns a set of features at each layer, and these features get more complicated as you move deeper into the network. And those features, the part of the power of the common nets is that they share the weights on features over space. We're working on an elaboration of this, convolutional neural networks, which we call OrbNet, which allows features to share weights not just over space, but also over orientations and scales. And this allows us to do two things that helps regularize the network and be more robust over fitting, allowing us to fit these more sophisticated models to noisy neural data. And it also allows us to potentially marginalize out things like scale and orientation and look for classes of feature tuning. And so this hierarchical model allows us to test potentially different hierarchical arrangements between visual areas, or even potentially between layers within visual areas. And this is all fit end-to-end, such that the models for the neurons in each layer, as well as the features in each layer, all fit simultaneously. So the way the model works for a given neuron is the features in each layer, the weights on them for a given neuron, are decomposed into an excitatory spatial set of weights and the weights on the features, and an inhibitory set of spatial weights and the weights on the features. This allows us to model the complicated context dependency that neurons seem to exhibit, which is what Michael is going to talk about next.
All right, thank you. So if we go back to these simple models that Michael and Saskia both spoke about, the way these models work is, of course, they describe some particular feature that exists locally somewhere in the image. So for example, with the kind of a standard simple cell response, if you throw it on a grating here, it should respond to this dark bar against a light background. It actually doesn't care about the rest of the grating. But if that's the case, if that's the way you describe that cell's response, it should equally well respond to this particular scene, right, where you have something that's dark surrounded by a lighter background. And so one of the things we can do actually with the Allen Brain Observatory is ask this question about how cells respond to different kind of stimuli. I'll try to, and so question one, just how do they respond to each individual stimulus? And you see per our definition of response, not all cells respond to every stimulus. In fact, there are about 25% of our cells that respond to none of our stimuli. And even then, you have things like static gratings where you get less than half of the neurons responding in any given visual area. Well, one of the things we can do is actually ask how cells' responses compare across different stimulus types. So here's a randomly picked experiment where we look at the activity of the neurons over time, aligned with the presentation of each of the different stimulus types. And you can see, hopefully, it's very clear that there are certain cells that respond to static gratings, right? These cells turn on when the static gratings are being shown, and then they turn off when the other stimuli are on. It's perhaps harder for you to see given the scale, but we actually have certain neurons that respond to the natural movies. We can tell this, on this scale because there are 10 repeats of the natural movie. So whatever feature is driving this cell, you see it 10 times, and there are a few of these cells. And so it looks like these cells are responding to a particular stimulus. And so we can ask how many cells are shared in common between the populations that respond to a particular stimulus. So for example, if I have some stimulus B that has for which 70% of the neurons respond, or to which 70% of the neurons respond, and some stimulus A to which 50% of the neurons respond, I can ask, what's the overlap of those populations? The maximum, of course, would be 50%. The minimum would be 20%. And say, for example, if this is a random label, if there's no relationship between the two, you'd actually get 35% if there's no information shared between responding from one stimulus to the other. So for that same experiment, if I plot the neurons and they label the neurons that are responding to the natural scenes, it's about 62%. For the natural movies, it's about 22%. And it just so happens that 62 by 22% gives you 14%. So the green cells here, yellow and blue makes green, are the ones that respond to both. Now, if you live in the land of simple cells and you've got some simple local feature that you're responding to, we would expect actually that these have perfect overlap, right, or at least large overlap. But in fact, this is at random chance. So if I tell you something responds to natural movies, that doesn't tell you anything about whether or not that cell responds to the natural scenes. So if we look at this for all pairs of stimuli, you see a kind of similar pattern. So dark red here is maximum overlap. And of course, every stimulus has maximum overlap with itself. And dark blue means minimum overlap. Most of these things are very close to chance. Chance is white. If I tell you something responds to locally sparse noise, you gain no information about whether it responds to any other stimulus. Remember, locally sparse noise is the dark and light spots. These are the things we thought would measure the receptive field, which would be a good first order description of the response. Instead, it tells us nothing about whether a cell responds to natural scenes or drifting gratings or anything like that. And similarly, if I say something responds to some movie, doesn't tell me anything about whether it responds to the drifting gratings, static gratings, etc. So this means that I can't have this picture for a standard cell that I look at this local feature and it tells me how it responds to this image. Something about this image is actually important, whether it's the fact that this is a stem of a flower, or I've got a ladybug here, or the entire context of the scene. It has to be important for the cell's response over and above just knowing about this local feature. So one of the ways of thinking about this is thinking about hierarchical computational processing. So put aside biology for a second, and let's grab a pre-trained convolutional network that's supposed to do image recognition. So in this kind of network, you see a systematic increase in sophistication in terms of this features that drive the units in the network as you get deeper in the network. So these are the things that kind of look like blobs and edge detectors. These are the things that are sort of the psychedelic 60s cells. And you've got some sort of mixture in between here where you have a few things that look gobblerish and a few things that look more complicated. Now we can ask the same overlap question of this kind of network. And in fact, what you see is early in the network, the things that look more like the standard picture that Saskia and Michael were describing, you have lots and lots of overlap, because if you have a simple local feature, if something is in a given stimulus set, it'll be in some other stimulus set, and it will drive that scene, or drive that unit's response. As you continue on, though, we get a larger and larger, we get to more of these complex features that are deeper in this network. You see a larger degree of independence closer to what we actually see in the Allen Brain Observatory. So this means we actually have to have a great deal of contextually driven responses in mouse visual cortex. They're much more complex and sophisticated than we actually expected from visual neurophysiology. And Dan's going to tell you about some mechanisms for generating context response.
So one form of context dependence that was discovered in visual cortex fairly early on is a phenomenon known as surround suppression that I'll describe now. Specifically, this is end stopping. So if we revisit once again our simple cell and we present bars of varying length to it and ask how well the cell responds. For a bar that's relatively short compared to the length of the receptive field, we get a fairly weak response. As the length of the bar increases all the way to the length of the receptive field, the response increases. Now we might expect that if we increase the length of the bar beyond this point, that the response will just stay at this maximum value because we're not changing any of the content of the receptive field itself. However, what is observed typically is that the response decreases. And if we continue to increase the length of the bar, the response of the cell will become very weak. This is a ubiquitous phenomenon known as surround suppression. Studies in mouse visual cortex have confirmed that excitatory neurons typically display exactly this kind of behavior in response to bars of varying length or gratings of varying size. You get exactly this kind of size tuning curve that I just described. Now, leveraging the power of mouse genetics to look at different cell types, it was also discovered that a particular type of inhibitory interneuron, the somatostatin interneuron, has a complementary size tuning curve in which these cells respond strongest to the largest gratings. Now, this was a particularly interesting result because it suggested a possible mechanism for how this surround suppression might arise. These inhibitory cells that respond to the largest gratings could be providing the suppression of the excitatory neurons at the largest grating sizes. So results like these around inhibitory neurons made us very interested and motivated to look very closely at the responses of inhibitory neurons in the Brain Observatory to our various types of stimuli. So here I'm showing the fraction of cells that respond to our drifting grading stimulus across all the different Cre lines, pointing out specifically our two inhibitory interneurons so far here. The somatostatin interneurons, indicated by red as I just described, indeed do respond very strongly to these stimuli. So almost 100% of the cells respond to these large grading stimuli that fill the monitor, the stimulus monitor. This is in contrast to the VIP neurons, which respond very weakly. Now, if we look at our natural scene stimuli, we see a very different picture in which both the somatostatin and VIP neurons respond very strongly. Another type of context is the behavioral context of the animal. So what is it doing? Again, inhibitory neurons display some very interesting properties. So in fact, some inhibitory interneurons track very well the running speed of the animal. And so in this case, it's the VIP interneurons that their activity follows very well the running speed of the animal, whereas other neurons don't seem to correlate nearly as well with the running speed of the animal. So we looked at this in the Brain Observatory using the models that Michael Oliver described, and we looked at how well the running speed of the animal predicts the activity of each type of cell. And in fact, we were able to confirm that there's a very selective effect for the VIP cells in V1 to be predicted well by the running speed of the animal. Interestingly, we found that when we looked outside of V1 in higher visual areas, this effect went away, and in fact, none of the cell types, including the VIP cells, were well predicted by the running speed of the animal. Results like these have led a model to emerge in the field in which context-dependent or contextual information modulates the interaction between an inhibitory microcircuit that is known from the anatomy between the VIP and somatostatin interneurons, and that this in turn can provide context by modulating the excitatory pyramidal cells. Now, in order to understand this, it's critical that we look at these two cell types simultaneously, because we believe that it's the interaction between them that has such a strong contextual modulation. And so we're doing this in follow-up experiments in which we are monitoring the activity of both VIP and somatostatin cells using a Cre-flip strategy to express GCAMP in both cell types, but distinguish them by expressing a red fluorophore only in a single one of the cell types, which are indicated with the red outlines around the cells in this video. So we are, during these experiments, presenting the wide variety of natural stimuli that Michael Oliver described, as well as monitoring the pupil and running behavior of the animals in order to really understand this context dependence. And this won't just allow us to understand better the mechanisms of context dependence in the inhibitory neurons, but also allow us to build better predictive models for all cells that incorporate context dependence.
So as you can see, this is a really large and rich data set that has a lot of possibilities for learning more about the computations going on in the visual cortex. And what we've described today is how we've been able to use this data to look at differences across the areas and how the information is represented throughout the cortex. We've looked at some of the interactions between the different types of stimuli and how the responses to these different stimuli really do vary for individual cells throughout the cortex, and then dug deeper into some of the local circuits that can be contributing to some of these types of interactions. And we're continuing to build on this survey data set by diving more deeply into some of the specific questions that have emerged from this large data set in order to kind of probe them a little bit more specifically with a little bit more ease of manipulation, and in a way that I kind of consider to be a little bit of a model of how we expect other people to also use this data to fuel their own ongoing experiments. So finally, I want to thank everybody who's contributed to this. We have a great team of scientists who've worked hard on analyzing this data, as well as a wonderful team of people who've worked really hard to collect this data and organize this data. It's a remarkable data set and a wonderful team that has done just a phenomenal job doing this. I want to thank everybody at the Institute who has supported us, and most especially our founder, whose vision and encouragement was critical to our success. Thank you.
We have just a few minutes for questions.
Are you able to use the pupil information to see what parts of the scenes are looking at, and has that made a difference in any of the analyses?
Yes, we have, for some of the data, we have decent eye tracking information, and it didn't make as much of a difference as we might expect. So, I mean, one of the biggest effects was there was a slight decrease in the complexity, but still, but it was still pretty high for most of the neurons. But yeah, it wasn't as big as we thought it would be.
Another question for Michael. In your Gabor model, after the linear sum of weights, is the last step you have a nonlinear threshold?
Yeah.
And is that fixed, or is that?
That's fixed, yeah.
I see. And have you thought about making that maybe activity dependent or not fixed, in other words?
Yeah, so I mean...
because that could take care of some...
What's that?
You train the bias.
Oh, I think he's talking about the shape of the nonlinearity.
You mean the bias or the shape of the curve?
Well, either way, you can change the bias, or you can change the shape.
The bias is fit, but the shape of the curve is fixed. It's essentially just a soft rectification. So yeah, I mean...
So the reason I'm asking
yeah.
is maybe some of the complexity could be captured by that. Is that possible?
No, not really. It's not really possible to capture this sort of complexity with just the output nonlinearity. And yeah, the shape of that can help a little bit, but it doesn't usually have huge effects on your model prediction performance.
Hey guys, that was really cool. So what the heck is going on with these neurons that respond to the gratings but not any natural scene? What is that? That can't be real. Like that. There aren't neurons that evolve to like just respond to gratings. Like this is crazy. So what's happening?
Yes, that's the question, right? This was a really unexpected phenomenon that we weren't expecting to see. We really thought if it responds to gratings, especially these full field gratings, we expected that something in these natural scenes is going to drive them. I don't have an answer for you, because that's kind of one of the, I think, questions that emerges. But do you want to?
Yeah, just to elaborate a bit. So I think this is why the sort of naive, though naive, the comparison with what happens in a convolutional net is interesting, because those are things that have an obvious task, right? They have to learn features to do this task. They show the same phenomenology in the sense that you get the same independence, and what's happening in those situations may not be happening in the mouse, but what's happening in those situations is you're developing a set of features that is very specific, right? As you move through a convnet, each successive stage has some set of units that are tuned toward whatever feature that's important for that particular task. If you think of something like MNIST, that's supposed to recognize handwriting digits, right? The last layer recognizes a 0, a 1, a 2, a 3. And then somewhere in between, there's some bizarre feature that's a combination of various things that is important for that task. but is not as simple as, say, a pixel. And in object recognition, a similar thing is happening, right? So presumably, I mean, one possibility is that the mouse visual system is sitting somewhere in the middle of some larger, well, it is clearly in the middle of some larger network that's developing features for some task. And because they're very specific, they show this independence property because you're essentially getting random projections in different, the stimulus types are essentially giving you random projections, which shows up as independence.
Yeah, can you contextualize a little bit the kind of finding that there's a lot of location specificity in these cells? You know, kind of going off the previous question where you have drifting gratings or you have the natural movie. So the mouse doesn't really have a sense of location of its place because it's just running on the wheel. So can you contextualize those new findings in context of our data?
I'm not sure I fully understand the question. I think the answer is no.
Yeah, that's what I was thinking. This is the Carandini and Harris paper.
Yeah. So we don't have a measure of anything like a location, right? There's no fiducial that we can say, oh, the mouse thinks it's here, whatever here might be, right? We can say how long it's run, but there's nothing to say where it started or what the topology in which it's running other than a hallway defined by the wheel, right?
Yeah. So we could possibly miss.
There's also no relationship between the stimulus and the mouse's running activity.
Yeah,
right?
that's what I'm asking.
And so without it being in a type of virtual reality type of environment, I don't know that you could speak to that at all with this type of data.
Okay.
Very, very nice data set. Have you compared OFIS with EFIS to see if the OFIS is detecting all the spikes, to see if that's one reason you don't see activity responses? And the second related question is, previously people have found in monkeys and cats and other animals that with natural scenes you get a tremendous sparsening of activity, but you also get a dramatic increase in reliability and precision of the responses. Have you looked at those type of things in your mouse?
Yes. So to speak to the first question about the relationship between calcium imaging and electrophysiology. So this is something that we've had a lot of conversations about, and there's a lot of work, and we're, as the EFIS, the NeuroPixel data set is coming online, we're starting to dig into some of these comparisons, but also Peter, who I see right there, had a poster yesterday that does show some nice work looking at what types of activity we're actually capturing with the calcium. So we know that we're not capturing single spike events in our calcium data, but we are capturing more of the burst-like activity, which then allows us to kind of more meaningfully relate the calcium activity with the EFIS, knowing those differences. And there's a lot of ongoing work within the team looking at those questions. I think it's a really interesting, questions. Regarding the second question, natural scenes, we are looking at the sparseness and the reliability. We do see, the responses that we do see, which we didn't show in this talk, but the responses are very, very sparse in response to pretty much all of the stimuli we show. And they also show a lot of variability. The trial-to-trial consistency of responses is not very high. I don't think that there are substantial differences across the stimuli, but I don't really remember off the top of my head right now in terms of that variability being less or more for natural scenes. But so we have been looking at this. We do know that the responses are very sparse, and we're looking at some of the things that could be contributing to that variability in these data.
Great. So thank you again to the Brain Observatory team.